Question 46. Question 46 asks that satellite RNA are present in. Now we should first understand what do you mean by satellite RNA. Sometimes plant viruses contain parasite of their own, and that is referred as satellite. Now satellite is highly depend on satellite RNA are highly dependent on their helper virus for replication and encapsulation. Now, so this plant viruses, they have a RNA that is known as satellite RNA. So is present in some of the plant viruses. Option D is the correct answer, that is plant viruses. Next is question 47. Question 47 asks that the one aspect which is not a silent feature of genetic code, not a so option A is degenerate. Yes, codon is degenerate as one amino acid code for several codon. So it is a degenerate. Option B, ambiguous. It is actually not ambiguous. It is unambiguous, and ambiguous in option, so it is incorrect. Why? Because codon is unambiguous, means codon is specific. C, universal, correct. Yes, it is a universal. And D, specific. Yes, it is a specific. So it is not ambiguous, it is unambiguous. It is unambiguous, means codon is specific. So B is the correct answer. Question 48. Question 48 asks that removal of intron and joining of exon is defined order in a transcription unit is called. So if there is a hnRNA, that hnRNA way some of the, that is from 5' to 3', then it has some of the exonic sequences, that is coding sequences. Some are intron, some are exon, some are intron, some are exon. Exon are coding sequences and intron are non-coding sequences. So here when introns are removed and exons are joined, this is mature RNA which is known as mRNA, and this phenomena is known as splicing. So splicing is the correct answer, that is option D. Question 49. Question 49 asks that which of the following is not correct for a molecule, not correct for a molecule acting as genetic material, not correct for genetic material. So now option A is given as it should be able to generate its replica. Yes, if it is able to replicate itself, then it is should be considered as a genetic material. Option B, it should be structurally stable. Yes, it should be structurally and chemically stable. Option C, it should be able to express itself in the form of Mendelian character. Yes. And option D, it should be heat resistant. There is no necessity of heat resistant. No necessity. So it is not correct for a genetic material. Question 50 asks that who developed DNA fingerprinting method? DNA fingerprinting, which is also known as DNA typing or DNA profiling, it was discovered by Alec Zeffrey. That is option B is correct answer. Question 51. Question 51 asks that the association of histone H1 with nucleosome indicates. Now when histone is associated to histone H1 associated with nucleosome means condensation is happening. So in this the DNA is condensed into chromatin fiber, then that's happen. So option B is correct answer. Question 52 asks that removal of RNA polymerase 1 from the nucleosome will affect the synthesis of. Removal of RNA polymerase 1. Now RNA polymerase 1 is involved in synthesis of 28S rRNA, then 5S rRNA and 16S rRNA, 18S rRNA. Now if we remove RNA polymerase 1, then there is no synthesis of these three. Means rRNA, ribosomal RNA cannot be synthesized, and in option E, ribosomal RNA, rRNA is equivalent. So removal of RNA polymerase will lead to the deficiency or inhibition of a synthesis of a rRNA. Question 53. Question 53 asks that which one of the following statement about the particular entity is true? We have to select a true statement. Now statement A is given as centromere is found in animal cell which produce ester during cell division. Actually centriole produce ester, not centromere. So it is incorrect. Statement B is given as the gene for producing insulin is present in every body cell. Yes, insulin gene present in every body cell. Okay, but the thing is that it not expressed in all cell. It is expressed in some specific type of cells. Okay, not expressed in all cell. So it is incorrect. It is correct that present in all cell. C is given as nucleosome is formed of nucleotides. Actually nucleosome is formed of DNA and histone, not of nucleotides, so it is incorrect. And DNA consists of core of 8 histone. Actually nucleosome has core of 8 histone, not DNA, so it is incorrect. So correct is B, that insulin is present, gene is present in all cells but expressed in some type of cells, not in all. Question 54. Question 54 asks that which one of the following palindromic base sequences in DNA can be easily cut at about the middle by some palindromic sequences, some restriction enzymes. So if we need to cut from middle, so if on both the strands, in the both reading strand, the same nucleotide sequence is there, and then only can cut. So if we go according to this, option C is most appropriate, as this have a sequence GAATTC, that is 5 dash to 3 dash, and 3 dash to 5 dash is CTTAAZ. Now here restriction enzyme can cut here, so both the cut is between TNA and TNA. If this cut here, then there is a possibility of a production of two GAACTT, that is 5 dash and 3 dash, and another cut they having a TTC AAG 3 dash and 5 dash. So in such cases it can happen. So option C is correct answer. Question 55. Question 55 asks that blank enzyme are used by every MacLeod and McCarthy in their experiment. Every MacLeod and McCarthy done a experiment after Griffith, transformation of Griffith, and what from S3 bacteria they have separated protein, RNA and DNA, and in this they have kept R2 bacteria. And here it does not show a transformation into the S3, it remain R2. It remain R2, but in DNA R2 converted into S3. So to confirmatory experiment to perform, they used different enzymes. So if we add DNAs in this, it will cleave DNA, and if DNA cleaved, and then R2 bacteria cannot be converted into S3, and this confirms that DNA is a genetic material. So they used like this proteases, RNases and DNases. So option A is correct answer. Question 56. Question 56 asks that one genome enzyme hypothesis was postulated by. One genome enzyme hypothesis. It is actually one gene, one enzyme hypothesis. And this one gene, one enzyme hypothesis was proposed by Biddle and Totem in 1948. And in this, they have stated that the gene controls metabolic machinery of the organism through synthesis of the enzyme. And this laid the foundation of biochemical genetics. So Biddle and Totem is the correct answer, that is option A. Question 57. Question 57 asks that identify the correct pair. Identify correct pair. So if we go with the option A, option A is talking about AUG and phenylalanine. Actually, AUG does not code for phenylalanine, AUG code for the methionine. B is given as UUU with the leucine. It is also incorrect. UUU code for the phenylalanine. C is given as UUA, and in front of serine is given. It is also incorrect. UA code for the leucine. And D is GUU, which code for the valine. So it is a correct match. So it is an identified correct match. Question 58. Question 58 asks that histones are rich in the basic amino acid. Histones are rich in basic amino acid that are arginine and lysine. So option A is the correct answer. Question 59. Question 59 asks that genes of interest can be selected from the genetic library by using. So genes of interest that can be selected from the genetic library. Now GeneBio's genomic library is a complete collection of cloned DNA fragments which comprise the entire genome of an organism. So we can elect a particular set of interests if we have a DNA probe. So DNA probe can help for this. So B is the correct answer. Why? Because our gene of interest we have to identify from this. Then if we have a complementary sequence which can be considered as a DNA probe. Question 60. Question 60 asks that a DNA replicates semi-conservatively. DNA replicates semi-conservatively and it was experimentally performed by Messelson and Franklin Stahl by using N14 and H15 isotope of nitrogen. So C is the correct answer.